Hello, and we're back with What Did I Do in Lab this week? Entomology Lab flavored, because that's the lab that I have this semester. So today was the first day of Entomology Lab, and we actually did like 3 different things. First thing we did, and I didn't get any pictures of this because I was paying attention, we were going over a bunch of different traps we can use to get bugs, and we did like a pitfall trap, a water trap, and a pheromone trap. Basically, a pitfall trap, you put a red solo cup in the ground, you put some water in it, wait for the bugs to fall in, profit. Kind of same thing with water trap, but it's above ground and a little bit larger. And then pheromone trap, you can buy them. You add water, they emit a bunch of different pheromones for specific insects, mostly like wasps and flies, so you have pests, etc., profit. That's not the interesting part. So the next thing we did in lab was doing some specimen collecting for 2 different things. So the first one we were collecting specimens for was a Wolbachia assay. So we will be finishing that in the next few weeks, but basically what that is, we're collecting different specimens of insects and then doing an entire multi-day lab testing for the presence of Wolbachia bacteria. So Wolbachia is a pretty common bacteria that is present in like over half of the different types of insects. I think it was like 60% of insects have Wolbachia in them, and basically we're just collected a bunch of specimens to see if there is the Wolbachia in there, which we will be doing in later labs, which I will be talking about. So most of the specimens were live caught, so you would just walk around swishing your little butterfly net around and then put them into test tubes full of ethyl acetate in order to kill them. This one specifically was a butterfly that I found on the ground dead already. That one was good specifically because you want to get the insects right from the net into the ethyl acetate, which is very tricky to do with butterflies because they're pretty large and they move a lot. So getting it one that was already dead, that already had enough DNA, was pretty cool. My professor said they've never done one on one of these butterflies before, so that'll be interesting. I ended up with 3 different tubes. Everyone was supposed to get 3, so we see the one in the middle, that's my butterfly that I found. The one on the right is some kind of little beetle, I'm not entirely sure. And then there's the one on the left, you can't see it, but there is a tiny little no-see-um at the bottom there. So basically what the ethyl acetate does, it serves a couple different functions. One is in order to kill the insect as quickly and painlessly as possible, which is what we always want to do. The second one is to kind of preserve the insect as best you can. And the third is in order to, this one is non-diluted, so it extracts the DNA, which will be used in later labs for the assay. So I will keep y'all updated on the progress of the Wolbachia lab, but that's kind of all we did for it for now. The other specimen collecting we did is for our big term project. So basically what we have to do to that is get 50 different specimens, so 50 different individual insects of, I think it's 12 orders and 20-something families. Basically, we gotta get 50 different bugs and hand them in at the end of the term, so we did some specimen collecting for that. In here, you can kind of see there's a butterfly right here. I'm not entirely sure what species, and then down there is, you can't really see it well, but that's the butt end of one grasshopper. I also got this big darkling beetle who took a really long time to die in the jar, and I felt really bad because he was just like running all over and like I felt really bad, but he did eventually end up dead, which is what we want. Yeah, he's a big boy. So my haul by the end of the lab section were 3 different things for the Wolbachia assay, as well as 2 butterflies, 2 grasshoppers, a big old blue bottlefly, and a really big beetle, so we're making progress. And this one you can see right here is one of the butterfly specimens. This is different from the one I just showed. It's a different species of butterfly. Now, we can't just keep them in the jar like this. They're not very easy to see, and it's also not super good to keep them soaking in the ethyl acetate for a long time, so we learned how to pin them to preserve them in our collection. So this poor quality video is of me trying to pin one of my grasshoppers while videoing, while trying to avoid being judged in public because I was sitting in the little atrium in the middle of the arts building, and there was like probably 20 other people in the room, but I had to finish pinning my bugs because I didn't want them like just loose rattling around while I took my box to class. You can also see there's only one grasshopper here, then the beetle and the fly. I haven't pinned the other grasshopper yet, and I actually don't have my butterflies on me. Right now, they are still back in the lab because they need to dry into position with their wings spread out. So sometime this week I'll go back into the lab and grab them and pin them into my box properly, but as of right now, they're just drying in the lab. We've also got my killing jar. This is just a mason jar with paper towel soaked in ethyl acetate. Basically, you just get the bugs in here, they die, and then you remove them, pin them, profit. This is my collection box. This is from the lab. I will have to return it at the end, but I want to keep my collection, so I'm probably going to have to order my own online, because I do have to return this one, but this is what we've got so far. We've got please focus, a blue bottlefly. He looked really blue while I was catching him, but he's kind of dulled out now. You can see that it's really, really fuzzy. We've got the darkling beetle. Got one grasshopper. I'm pretty sure this is a red leg grasshopper due to the red legs. And then another larger grasshopper. I don't know what species this is. It had really pretty red wings, which is kind of why I caught it, even though I already had a grasshopper. Then, yeah, I've got my 2 butterflies that are gonna come in here. I think this weekend I'm going to be going out to the creek and seeing what else I can get. I do need to buy a butterfly net, just because I need a way to catch them, and without a butterfly net, it's just way harder. So I will probably make a video about that, showing off my haul. But yeah, that's what I did in lab this week. Behold my bugs.